With us today Professor L, the University of Estonia, University of Tartu, Estonia. Professor Kasik is an internationally renowned researcher whose work focuses on understanding the molecular mechanisms underlying neurodegenerative diseases, including Alzheimer's, Parkinson's, and Huntington's disease. His research aims to identify critical cellular pathways and potential drug targets to develop effective therapeutics for neurological disorders. With extensive expertise in intracellular quality control mechanisms such as autophagy, mitophagy, and ERphagy, Professor Kasik has made significant contributions to both basic and translational neuroscience. We are honored to have him with us today to guide us through this course and share his insights on neurodegeneration and drug development strategies. Now I request Professor Ellen Kasik to address the audience.
Thank you. You can hear me well. Okay, so I'm happy to be here. First, I'm honored to be here. Thanks to Danzir Organizers University. And it's, as it was told, my first time in India. So everything is quite new for me. Yes. Okay, now it's better.
Sir, you have a caller mic.
Yes.
It's not.
Ah, I see.
Maybe this turned off.
It's green.
Yeah,
It works.
perfectly fine. Now you don't have to stay around.
Okay. So you are probably wondering from where exactly I am. So you know something about Estonia? Probably not. So we are located between Finland, Russia, Sweden, and other Baltic states in the north of Europe. The university. Yes. The university I'm coming is actually one of the oldest universities in Northern Europe. So it was established 400 years, almost 400 years ago by King of the Sweden. Estonia was then under the Swedish rule before the Russians came. And since then it has been quite, quite good university. And it was one of the best universities also in Russian Empire. So it is old picture. Now it is looking like that. Then I'm also from Department of Pharmacology. This is actually one of the oldest departments, or even the first departments of pharmacology that was established in the world. And we have the founders of pharmacology, Buchheim and Smedeberg, who came and established this department. So Buchheim was the scientist who transformed this traditional materia medica into this modern drug classification system. Before that it was just a text showing that these compounds or plants are doing that and that, but after the Buchheim, pharmacology became really classification of drugs. And also his student Oswald Smedeberg was first to establish the pharmacology itself. So before that there was almost no pharmacology. After that the pharmacology became independent discipline. So then when they became famous, they moved to Germany to better universities. So this is about the history. Now we are located in the new buildings. You can see the biomedicum where we are it's the institute which includes all these preclinical departments, including the pharmacology department. And this is the main university clinical department building. But okay, this is about our university. So we are starting the course with a general introduction to neurodegenerative diseases, about symptoms, about mechanisms, not too much mechanisms, and then about the drugs, a lot about the current drugs. What is your background, educational background? Biology, biochemistry, chemistry, biomedicine, medicine, something. Pharmacology. So all you have pharmacology background or biology? Okay. So when I started, I was a biology student, and I was in department of biology, and we cared only about the mechanisms. We did basic research in the lab, and I did not know much about patients, much about symptoms, nothing about the drugs. And it was a mistake. I think my education was not good at all. So then, as a PhD student, I moved to the medical faculty. And my supervisors in Estonia and Helsinki were all medical doctors, also working part-time as a medical doctors. And then I discovered that it's really not enough. Even if you study one molecule, you are testing it in hex cells. You should still know what you want to do with that molecule or compound. What is the disease you are really studying? So I started to learn, to understand more about medicine. So I made a master in in medicine, and PhD was very medicine orientated. And why it was good? You may think why I should know about the drugs they use in case of Alzheimer. It is good because you should, if you develop something, you should know what is the real need for that drug. So when we are now studying the rare Wolfram syndrome, it is maybe 2 per million is the prevalence, which is very small, rare disease. We are looking for the drug candidates. Then we are not doing it just in the lab. Okay, we work very simply with cells in multiple plates, but we have a constant contact with patient societies. So we visit the patient society meeting in somewhere in UK where they have more patients once per year. We have a contact with the smaller drug companies, okay, not at the highest level, but at the people who are developing the drugs for these diseases. And the reason is that we need to know exactly what patients need, and we need to know what the drug companies are doing. It's of course not the newest information, but still it's more new than in publications. And why we need it? We need it for two reasons. First is that it motivates ourselves, because in an academy, while we are doing research, what is the motivation? For younger people it is career and so on, but for me it is not motivating anymore. Only thing, money is also not motivating, because in academy you don't make money. Only thing that is motivating is that you will find something that is good for patients. Or even if what you are doing is not going directly to patients, somebody else is developing your knowledge further to something which helps the patients. So that actually the patients, this is the main motivator, at least in pharmacology. And another thing is that if you know all these details, what is needed, what patients want, then you also know what the funding agencies want. Because, for example, what is the problem when we evaluate the grants of the European Health Agency? The aim of the grants is to develop new therapeutic modalities, drugs, and so on. Then they expect something which is immediately, or at least after some delay, useful for patients. If you are doing basic research, you don't get any money. So to get money, you should know again the need very much. If you develop something, you should know the need, and this helps you to get the grant money and to do more work and better work. But okay, so the idea of this talk is that if you study some compounds in the lab, and even if you are chemists, you should know the disease, what for this compound will be developed very well, and you should know the current therapies. So I think I will start with very, very general explanations. What are neurodegenerative diseases? What are the factors making the neurons very sensitive? And then go to diseases. So I don't want to be the only person who is speaking. So I will try to ask questions from you. And so if you have questions, then after each block, I have also a small, break for questions, because at the end we don't remember anything what was at the beginning. So please be free to ask after these blocks whatever questions or specific or generic you have, please ask. Sometimes I don't know what to tell you, but sometimes I may know that. So first is that how you define neurodegenerative disease. So this is question first for you. For you, what is neurodegenerative disease? Please.
When there is a structural degeneration of the specific neural components, for example, in Alzheimer's, where there is neurodegeneration of the hippocampus region of the brain and the prefrontal cortex too, we classify it as a neurodegenerative disorder because some of the structural components that are responsible for the normal neural functioning are getting degenerated.
So this is what you think is the neurodegenerative disease really existing in the term neurodegenerative disease? Is it existing in clinical settings? No. There is no really disease called neurodegenerative disease or disease group called neurodegenerative diseases. So that the definition is actually mostly for basic scientists and for the general public, that neurodegenerative diseases is disorder where we have loss and function of neurons, and this happens somewhere in specific area in brain, and after that neurons die, brain will degenerate. And there are some core characteristics. So first, that it is always progressive. So you have always more degeneration. Slowly, more and more from the brain will degenerate. Then it's usually selective. In some place, it starts from specific place, with exception of maybe prion disease, and then it is becoming, the area which is affecting is coming bigger and bigger and bigger, and it may end as Alzheimer. It may end that you have in all brain areas degeneration. And then the third characteristics is that neurons are dying. So they are dying, and if you have enough neurons that will die, then you will see that there is brain atrophy, or in case of Parkinson, loss of dopamine, dopaminergic neurons. It is visible after some time if it's strong enough, this degeneration. And the fourth thing is that you don't get these neurons replaced that are dying. So who dies, and that's it. So this is the general, characteristics. Then what you see at the level of the tissue or at the level of cells, first is that not always, but usually you have a protein misfolding and aggregation. It's not 100%. There are some examples where you really don't have it. Then another thing is that you very frequently have a mitochondrial dysfunction and oxidative stress. Mitochondria do not work anymore properly, and mitochondria are main source for the free radicals. And if they don't work properly, then you will have more free radicals and oxidative stress. And oxidative stress is not just because of mitochondria, this is just one reason. And what is now very popular is that you have also inflammation, neuroinflammation. In most cases it is secondary to the damage. What happens when neurons are dying? What happens with this content of neurons? It will be going back to the brain, it is everywhere. If it's apoptosis, then they will die more slowly, of course. But anyway, there is something which induces inflammation. Sometimes you have inflammation also as a reason. So this is just an image to show you that different diseases start from different places. So let's start from Parkinson's disease. And Parkinson's disease starts from the midbrain, and at the cellular or histological level, you will lose first the black pigmentation in substantia nigra, which means dopaminergic neurons are dying. And then you have a Lewy bodies. Not always, but you in most cases have them. It's midbrain, striatum. Then the ALS, amyotrophic lateral sclerosis, it starts from brainstem, motor areas of cortex and spinal cord. So a little bit from cortex and a little bit from the brain stem. And it is related to motor neurons. Motor neurons will die. What you see at cellular levels, you can sometimes see some inclusions. They call it Punina bodies or Lewy body-like bodies. Then Huntington disease starts from the basal ganglia. At macroscopic level, there is atrophy, neostrial atrophy, neurons die, and you will have inflammation, astrocytosis coming, so on. So, frontotemporal degeneration or dementia and Alzheimer's disease. So they are in some sense similar diseases. They start from cortex, from specific cortical areas, different areas. They will lead to atrophy, and they have deposits. Alzheimer has beta amyloid deposits, tau, tangles. Frontotemporal dementia has also tau deposits or big bodies. So this group is little bit overlapping. And then you have a rare disease, very rare disease, prion disease, where you have everywhere in cortex strong atrophy, spongiosis. The brain will be at the end like a sponge. So I have spoken with some neurology surgeons or neurosurgeons who have seen this, and they told me that at the end the brain, after they make the pathology to understand what is wrong with patient, then the brain looks like sponge. So a lot of diseases, but there is no generally accepted list of neurodegenerative diseases. So the main and the best classification of the diseases is this International Classification of Diseases 11th revision. And also if you check the World Health Organization disease classification or lists, they don't have neurodegenerative diseases. And the reason is that in these clinical settings, these diseases are quite differently managed. And they are clinical syndromes, they are not together. There is now nothing like neurodegenerative disease. But anyway, you should know how to call these diseases. So why it's relevant, why you should know that. So it's this is just a general homepage. If you go from here, this classification browser, then you will get here diseases. So there is list of disease groups, and 08 is the list of the diseases of the nervous system. If you go in, you will get here a lot of other, how to say, subgroups. For example, if you go to movement disorders, then you will get the movement disorders, and here you have, for example, Parkinson at the beginning, A800 Parkinsonism. Or you have here Lewy body, the Alzheimer's disease, and then these motor neuron diseases are not so close. So why this classification is relevant? Usually it's not so relevant, but sometimes it is. So for example, this funding agency, European Health and Digital Executive Agency, is giving grants. And it is giving grants specifically to find new therapies for neurodegenerative diseases belonging to group 08. Some people, basic scientists maybe, they don't understand what this classification is. And they are applying, asking money for diseases which are maybe belonging to group, I don't know, 17 or whatever. You have some neurodegenerative component having diseases also on the other groups. So that if you are speaking about disease, then okay, it's good to speak about neurodegenerative diseases, but for clinicians this is not disease. For clinicians and for these clinical orientated funding agencies, you should have real disease or disease groups which have the codes. And I don't know how about it here, but also in Europe, at least in clinics, if you put the diagnosis, you need a code. The code means that the health insurance fund will give some money, and so on, so on. So you don't need to know these codes, but you need to understand from where they come and how to find them. So next, that what kind of diseases we have. We all know there is Alzheimer, we know there is Parkinson, then we know there is Huntington. There is also multiple sclerosis. There is also the SMA, ALS. Then how frequent they are. So the list is not so short. You have actually much more diseases. So I listed here most frequent, most prevalent one. So how prevalent they really are. So Alzheimer is really prevalent. It's difficult to say how many patients we have per world in world, maybe 50 millions. The statistics is quite complicated to get. So I have therefore US statistics here because they have really trustable numbers. So we have 7 million Alzheimer patients at the moment in US. And as an average, after the diagnosis, as an average, the life expectancy is 4 to 8 years. But sometimes people can live 10 to 20 years also because the disease is very heterogeneous. Sometimes the progression is very slow. Then you have Parkinson's disease, 1 million patient at the moment in US. So 7 to 15 years after diagnosis, but sometimes with good therapies, good care, it's longer, sometimes even near normal lifespan. Then you have multiple sclerosis or sclerosis multiplex, 1 million patients in US. The therapies are okay there, so I mean 25 to 35 years life expectancy, not so much shorter than normal life expectancy, but here the drugs are an issue because if you take, I don't know about the drugs here, but if you take the cheaper drugs, they are not so effective. If you go to expensive antibody-based drugs, they are so expensive, not everywhere you can afford it. They are working better. Then amyotrophic lateral sclerosis. It's already relatively rare. Approximately 25 plus thousand patients in US. Life expectancy up to 5 years. But some people, a small fraction, disease is very heterogeneous, survive more than 10 years. Then you have Huntington disease, 30,000 patients, 10, 20 years after diagnosis or symptoms start. And then you have spinal muscular atrophy, SMA, 10,000 patients. It's very variable. It is type 1, type 2, type 3, type 4. Type 3 and 4, they are living, life quality goes down, but they don't die. And the type 1, they will die if they don't get the treatment during first years of their life. So then we have a lot of diseases, huge amount of diseases that are less prevalent than that. And then one example I included here because this is such kind of classic disease is the prion disease. In States they have maybe only 300 patients, but the life expectancy is very short because the degeneration, accumulation of the agent is very fast. So it's such kind of disease everybody should know. So about the frequency, as I told that it's really difficult to get the frequency. You can't compare different countries because the statistics is different, the diagnosis is different and so on. So if you think what is about India, because I have all statistics coming from US or from Europe, is it also similar to US values? So it's not exactly. Some disease is less prevalent, some diseases are maybe more prevalent. And US and Europe, they have old population. India has young population. How to take that into account? But if we take, for example, Alzheimer, the graphs are different, but this is just one graph I found from internet. This shows that the prevalence in India, Alzheimer prevalence in India, normalized to the age, is a little bit less than in Europe, not significantly. So Alzheimer is quite similar. I think all these other diseases more or less similar, less frequent slightly, but in real life they exist. Some diseases, MS is significantly less prevalent. Sometimes the differences are very big. For example, Alzheimer, the blue is relatively lowest prevalence. So it's Europe, India, part of the Africa, part of the South America. Then you have also some countries, North Africa, Turkey for some reason, where the prevalence of Alzheimer is very high. So I'm not completely sure whether this is correct, because you can have these statistics in many different ways. So there is heterogeneity, different areas have different genetic backgrounds. So this part was then about neurodegenerative diseases in general. So what do you think about that? What you can ask? So I need some input, please. Stroke. So stroke is not neurodegenerative disease, but it has very strong neurodegenerative component. Because if you have some stroke somewhere localized, then okay, there is no. But okay. Yeah, if you have something, then it will be good to have later. But it's okay. I will. It's okay. Don't worry. So in case of stroke, you have always this core area where you don't have the blood flow. Why stroke happens? There is no blood flow in some area. This specific core area dies away anyway if you don't restore the blood flux quickly enough. But then this core area is surrounded by penumbra, if I pronounce it correctly. And this penumbra is, for this penumbra there is question, will it die, will it not die? And sometimes it is dying slowly, it degenerates slowly. So it's, you can say that this penumbra degeneration is neurodegeneration. But this is how to say, there is little bit of neurodegeneration, it's not neurodegenerative, yeah. It's quick, but this penumbra is dying slowly. Okay, it's not years, but it's not half an hour. And sometimes, actually this is one of the main questions if people have a stroke, that how to keep these surrounding areas of this core, how to keep these surrounding areas healthy. So, any more questions about that? Okay, we can go on because it was very beginning. So, why the brain is so sensitive? Please tell me. Why the nervous system, neurons are so sensitive, why they want to degenerate. Your fat cells don't want to degenerate, or my fat cells at least. They don't go away anyway. Why neurons are sensitive? Just, how to say, what you think? Yes, but also your kidney has very high metabolic demands. Kidney has much higher metabolic demand than oxygen, but kidney has also very high oxygen demand. But this is good reason. This is one reason. What else? Yes, this is important. There is no regeneration. What else?
So they don't divide. Once they are formed, the number only decreases with time. It isn't
Yeah, no
increase.
regeneration. Yes. And what else? So you have already two important points. Or three even. Okay. So I tried, it's my subjective listing why the nervous system is so sensitive. It's most complex. You don't have anything else in the body that is so complex. Then its development is very complex. You don't have such kind of cells in the body. If you take the motoneurons, where end of the cell is 1 meter from the soma, energy demand was already, how to say, said. Then there are neuromuscular connections, axonal transport, myelin, axonal signal transmission, and they don't renew. Let's start from the beginning. So the nervous system, first, you probably know that, but maybe somebody not. The nervous system is not only brain. The brain is the central nervous system. So the brain and spinal cord together is central nervous system. In addition, there is also peripheral nervous system. So all these nerves are going to somewhere in periphery, and you have also the central, how to say, cell bodies, somas, neuronal bodies, somewhere in periphery. So if you take, for example, enteral neurons, you have a lot of neurons and neuronal bodies to your stop. So it's complex. It's very complex. What do you think, how many cells, or better to say, how many neurons we have in brain? Just what you think, approximately. Million, billion, trillion? How much? Billions, yes. So it's billions. So it's actually, as an average, 86 billion neurons in brain. Then there is 1 million in the spinal cord, and not too many, just 0.1 billion in peripheral nervous system. Not really small number. Then, in addition to that you have a number, you have 80 and something billions. You have the variability. So it's not only neurons in the brain. If you take the brain, then you have half of the cells that are neurons, and then another half or more that are supporting cells, not neurons. So you have very different cells in nervous systems which are specialized to something. 3,000 cell types, or 3,300 currently. So they are all doing something different. So this is just a figure showing you that you have different neuronal subtypes: enteric neurons, parasympathetic neurons. Some subtypes are very rare. If you take neurons responsible for some kind of specific sleep. You have maybe 1,000 of them only in the brain. So some are not very abundant. Okay, you have 86 billion neurons, but these in brain, these neurons are doing something. They are depolarizing, they are passing the signal to another neurons, and they use synapses for that. So synapses contact between the neurons, between the axons of neurons. But you think how many synapses we have in brain? So more probably than that. So as an average, it's a very average. We may have more than 100 trillion synapses. So it's more than 1,000 synapses per neuron. It's a very average number. Neurons are very different. So this is, okay, they are just numbers, but what people are now doing, they are trying to make a map of the brain. So if you take the brain atlas, then these brain atlases are usually, how to say, looking something like that. So you really have the general structures, but in each structure, if you take the cortex, for example, you have all these variability, and this atlas is showing you really nothing very much. So people are trying to build up the real brain atlases. So this is possible nowadays, and it requires two things. First, it requires really high resolution imaging. It's the electron tomography and such kind of imaging techniques. And another thing, it requires the computing power. So therefore, a lot of these leading laboratories in States, they have teamed with IT companies, Google and Harvard, and it's not the only team, and they're trying to produce such kind of neuronal maps. So it started, I think, approximately 5 years ago, and now the first publications are coming out. What is the point? Why they want to do it? Scientists are doing it because they want to understand what happens really, how signal is moving, how brain is working, and the IT companies, they are hoping that this is useful for them for the artificial intelligence. Plus they are just supporting science. So the Google, Meta, they have invested a lot of money to build such kind of systems. So actually the computer power is now not limiting. What is limiting is the imaging. You really can't image too big pieces of brain. So at this current situation, they have such kind of, I think, Drosophila brains fully scanned, and they have mouse brain, pieces of mouse brain, few millimeters, cubes scanned, not the full brain. The first images are published. So here you will see one example where you have white is the neuron bodies, axons, neurids going of that specific neuron. And the green, blue is neurids, axons coming from other neurons. So you see that single neuron is surrounded. You have here some kind of supporting glia, whatever cells are surrounded by huge number of neurids coming from other neurons. And what you can see here, but there are images also where the synapses are shown. So most likely these axons are full of synapses. There should be, I don't know how many hundreds of synapses also here somewhere. what they are doing, they are also counting the synapses. They are counting, they will find out where is the synapse. They are counting the synapses and they are trying to predict how signal is moving. What is also interesting, green axons are excitatory neurons, means that they will activate something. If this green neuron, a neurid for example, is having synapse with a white one, this means that it could activate the white neuron. And the blues are inhibitory. Blue means that if the signal is going on from blue, it is trying to suppress the white one. So that there are excitatory neurons and there are inhibitory neurons. There are a lot of excitatory neurons, there are not so many inhibitory neurons. So this is one neuron. Here is the big piece. So it's the, if it's correct, then it's the cortex, small piece of human cortex, and they did the same here. Here the color means just that this is the cortex starts from there, the first layer of cells, and so on, so on. So you see the structure is complicated, and from here the neurons are giving axons to other structures. They excite these structures, these neurons excite these neurons, and so the signal moves. So complicated. But then if you take the brain, it's not coming from nothing. There is no brain and brain. It should develop something. So the development of this system is even more complicated than the system itself. So it starts very simply from the neural tube. Just a tube, few cells, they will have a tube, and then after that, from this tube you will get the precursors, and then the precursors will start to proliferate. So you have a lot of neurons. So it's at the beginning of the development. So after that, you have your, how to say, young neurons. These neurons are not where they should be. They have to migrate to the place in developing brain. They have to find, they are coming from this tube, they will have to find more or less correct place in the brain. This is okay, but then after that they are just the neuron bodies. After that, the axons and neurids should grow up from this cell body. It's the neurid growth, axonal growth. Then you will have axons, then the axons should form the synapses. And then only after that you have the real brain. So this development of nervous system is most sensitive and complex part of this. So it's also most sensitive to drugs. So if you take, so you have also, if you buy a drug, you will get it from the pharmacy. Then it's accompanied by this leaflet. Is it also same here? You can read from this leaflet what were the side effects. Can it be used during the pregnancy? So what is mostly written there? Can it be used during pregnancy? What is the comment there? Most usual, we don't know that they don't know that. A very small part of the drugs could be are not having risk during the pregnancy and are tested to be safe. In most of the cases, there is no clear evidence whether it's safe or not. And one thing why these drugs are not safe, one thing they are not tested, is that since in the uterus the developing nervous system is very sensitive, all these side effects which are not affecting adults or older children very much, they may be toxic during the development process and during the nervous system development process. And to minimize that also, this, if you do toxicology testing, I mean preclinical toxicology for FDA, and you have a drug affecting nervous system, then you always should, in addition to neurotoxicology, you should do these developmental neurotoxicology tests. And the reason is just that this is most sensitive system, most sensitive time, and the minimum side effects could affect this process. You will get different brain. So it's just one example, that illustration. So one of the possibility speculation is that it's clear that autism spectrum disorders, autism, is related to brain development. And it's already related to brain development in uterus. And one of the, results people have demonstrated is that at the beginning in brain development, the cortical area of the future autist children is bigger. So when brain is developing, then they have, at the beginning, these future patients in uterus have more neurons in cortex. They will get the development faster. At the end, when they are older, the number becomes same, but at the beginning in uterus and first years, they have more neurons. And this is somehow related with autism. It's just one speculation. Then another thing is that when axons are growing, how they are growing. Axons are growing, and then there is pruning, axonal pruning. So if, for example, you have in developing nervous system, you have neuron body in the brain, and it should go to periphery, it's motor neuron. It should find the target somewhere here. So it's, even if you have a developing organism, it's a long way to go. So they have these axonal growth cones. These axonal growth cones are going through the tissue, looking for the right place. There is some chemical gradients directing it to this correct place. And then a lot of axons are doing it. So you have at the end too many axons, and then some axons are dying away. So at the end you should have a normal number of axons, as much as needed. So this process is, okay, I was very generic here. This process is also very sensitive to every kind of damage. So the energy demand that was mentioned here. So neurons have very high energy requirements, and they depolarize, repolarize. They have long axons, things should be moved through this axon, takes energy. Everything takes huge amount of energy. And this energy is coming only from mitochondria. There is almost no hope to get much energy from glycolysis or from fatty acids. Neurons will get it only from mitochondria, mostly from mitochondria. Mitochondria need oxygen. If you have no oxygen, mitochondria can produce ATP. In case of stroke, this means that no ATP neurons will depolarize, die. So another thing is that, okay, they need a lot of ATP, but they need it when they need it. So this energy, there is no, storage for energy in neurons. This energy has to be produced at that specific moment where it is needed. So the supply and use should be very much close to each other. And because of that, if anything happens with energy production, anything happens with mitochondria, then the nervous system is immediately affected. Mitochondrial defects, lack of oxygen, toxins. So neurons have no energy reserves. Whatever affects mitochondria leads to problems in neurons. This is just a figure to show how these neurons are using energy. So 1/3 is going to just housekeeping, and then these 1/4, 75% is going to this action potentials, synapses. This is RP. Is it releasing of something, or is it restoration? It's probably restoration of membrane potential. So the action potential and its restoration takes half, and synapses, the release and uptake of synaptic vesicles, takes another half from that part. If you take normal cells, not muscle cells, then they have much less, they have only housekeeping to do. Then neuromuscular connections. So these are sites where neurons, motoneurons, connect with skeletal muscle fibers. So in the image you can see this is the motoneuron coming, it's motoneuron fiber. The motoneuron fiber is giving branches, and these branches are going to muscle. So it's the, neuromuscular junction is this place here. So the pocket-like structure at the end of the axonal tip. And these neurons here, they are releasing, if activated, acetylcholine to this pocket or synaptic cleft. This acetylcholine is binding acetylcholine receptors that are in the muscle fiber membrane, and then these receptors trigger the depolarization of muscle membrane. Depolarization of muscle membrane leads to the calcium influx. Calcium induces the muscle contraction, and so it works. Neuron activates, muscle contracts. And these neuromuscular connections are very sensitive to damage. So if you take the toxins, then most of the deadly toxins in nature, they affect these neuromuscular junctions. What do you think, what is the logic? Why most of toxin, not all, but big part of toxins do it specifically that? What happens if you block the neuromuscular junction? Paralysis, yes. So you can't move anymore, and this is the aim of the toxin. If somebody wants to eat you, you should stop it doing that. So many toxins, very strong toxins, cone snail toxins, are doing that. They are blocking the acetylcholine receptors. Or one example, good example, is curare. curare is actually in some kind of plants, and the local Indians, they just use these plants to do the fishing. So if this curare was going to the small lakes or small ponds, it was killing the fish, and you made good fishing. Then there is axonal transport. As I said, you take the motoneuron, you have in brainstem, you have a soma somewhere, and then it's 1 meter to go to somewhere to move your leg. And the axon, this 1 meter long axon, should move all the stuff from the body to the end of the axon and back. All the synthesis, not all, most important synthesis happens in the body, and all these compounds, building blocks, they should go to axon somewhere, somehow. And then if these building blocks or compounds become old, not compounds, sorry, proteins become old at the end of axons, they should come back to the body. If this is not happening, then the axon will die away very quickly. So there should be very efficient axonal transport. So this axonal transport is essential to move everything, molecules, organelles, proteins, between the cell body or soma and synaptic terminals or presynaptic terminal. So there is anterograde transport and retrograde transport. Anterograde means that everything goes from body to the axon, and this system uses the motor proteins called kinesin motors. And then there is retrograde transport that carries old stuff back from the terminal to body, and this retrograde transport uses tunane motors. So kinesin is single protein, tunane is huge complex of different proteins. And these motor proteins, they should move somehow. So we have here just the cargo and motor protein. They should have some kind of railway. So there should be railway going through from the soma to the axonal terminal. And this railway contains microfilaments of actin, microtubules, different tubulin, and a lot of neurofilaments there. So actin, tubulin, neurofilaments, and there is tau related to Alzheimer. This tau is also somewhere there. So there are some toxins or drugs affecting and using axonal transport. So if you take the cancer drugs, Taxol, colchicine, vinblastine, nocodazole, they are all affecting this railway somehow. So Taxol makes it more stable, then colchicine makes it less stable. You have problems with tubulin polymerization. Vinblastine depolymerizes tubulin, and nocodazole as well. So that if you use these drugs, your tubulin network is, too strong, which means it's messy, it's not anymore nice railway, but it's like that. Or it's with holes if the polarization is not enough. So this is aim actually why they use this drug in cancer treatment, because in case of cancer this blocks mitosis, cancer cells do not develop. But all these drugs affect also axonal transport in the brain. So we are using sometimes them to affect axonal transport. So it's such kind of side effect at the level of brain. Or another good example is the neurotoxin, tetanus toxin, produced by Clostridium tetani. So it is such kind of two subunit big protein, 50 kilodalton and 100 kilodalton heavy chain protein. So it's so big that it is not going to brain. Why some compounds and big proteins are not going to brain? There is barrier. So it can't go to brain because of that barrier. So it has therefore such kind of clever way. So if you take the animal, for example, animal injures somehow its leg, and then this tetanus toxin is going to the leg. In leg you have muscles, but you have also the terminals of motor neurons. So it will be internalized to motor neuron, and then it uses the transport. Is it, what do you think? Retrograde or anterograde transport now? Retrograde back. So it uses retrograde transport to go back to the body of this motor neuron. And then from this body of motor neuron, it is going externalization, internalization, it is going to inhibitory interneuron. What this then happens? So first is that it blocks the signaling. It cleaves the synaptobrevin, the protein here that is responsible for releasing of the neurotransmitter. If you don't have neurotransmitter release for inhibitory interneuron, what the motor neuron will do? So inhibitory interneurons, they inhibit other neurons. If they don't inhibit, then other neurons become too active. If the motor neuron comes too active, what it is doing with muscle? There is contraction, yes, exactly. And this uncontrolled contraction is the main symptom of the tetanus toxin poisoning, not poisoning, but this tetanus, main symptom of tetanus. So it's a very clever system how the toxin is using the retrograde axonal transport going so far from the initial site of entry, and how then it affects back to this initial site. Then there is myelin stealth. So this is what you know about myelin stealth.
Property of the neuron that help in circulatory conduction.
Exactly. So this is such kind of lipid layers, multi-layer lipids, which are surrounding not all but some axons. Not single axon, but usually axonal bundles. So there are more than one axon inside, if I believe it. I think it's correct, yeah. And this myelin stealth is produced in peripheral nervous system by Schwann cells and in central nervous system by oligodendrocytes. And what for it's good? As you told, it's to increase the speed of the nerve impulse movement. So that when usually the signal moves from one side to another, this requires depolarization of axon. And depolarization itself is a little bit slow. Okay, it's fast, but it's not super fast. So if you have just the axon and a lot of these channels here, this is making the signal a little bit slower. And to make the signal faster. Some areas are just covered by myelin, and through these areas the signal goes through very fast. And between these myelin stealth you have these areas, small areas where you don't have myelin. They are called node of Ranvier. So if you take multiple sclerosis, then you don't have so much myelin, which means that signal conduction is slower and you will have problems. Just one example. It was in 50-60 years ago, at least in States, they were selling hexachlorophene as an anti-infective and antibacterial agent. It was used in soaps and in skin cleaning, skin cleaners, also in agriculture. And some of these were used for babies. So babies were taking bath. People were thinking that it's better if the bath is containing something antibacterial, and they added this hexachlorophene containing things to the bath water. But what was the problem is that this compound was very lipophilic. So from the water it absorbed through the skin very quickly. It had no problem to go from skin to body to nervous system. And it's somehow, I don't know exactly, it uncoupled OX-Foster, and it induced brain problems, which was the loss of the myelin, edema, vacuole formation, and at the end it led to axonal degeneration and so on. So when they discovered that out, then it was discontinued. But one thing is that for some reason the myelin, it was demyelinization. Okay, and then axonal signal transmission. So we talked about that a lot. So signals should move from one place to another. This involves axonal action potential, which means that at some point the sodium channels open, you have the polarization, then the potassium channels open, you have repolarization and restoration. So, and if it's triggered, then the signal starts to move from this place where it's triggered further. So it's the signal transmission, simplified version from that. There are again some toxins affecting that. The best known is tetrodotoxin. So it's such kind of interesting agent found mostly in pufferfish. And it inhibits the sodium channels in periphery. So it can't go to brain because of the brain-blood barrier. It's slightly too complicated molecule for that. And it blocks the sodium channels in peripheral nervous system. What happens if you block the sodium channels in periphery, in peripheral nervous system? What happens? What you think? Sorry? Yes, but what happens if you think about action potential? You don't get action potential because the sodium channels are inhibited. if you don't get action potential and signal transduction in periphery, what happens at the level of body organism? So at the beginning it becomes numb, but after that you don't have any more nervous signals everywhere. You have a paralysis, you can't move anymore. You can't respire anymore because respiratory muscles here, they also want signals from somewhere. So everything stops in body, but the toxin is going to brain. Brain is, at the beginning it's okay, but of course if there is no blood anymore coming in, then the brain will die because of the lack of blood. The fish itself contains it in huge amounts, but fish is very clever. Fish has this specific sodium channel mutated, so the compound is not affecting the fish. So it's famous toxin. Why it's interesting? Interesting is that people are eating this fish a lot in Japan, at least. So this is so popular that they don't have enough in sea, so they are also farming it, and they are eating it. Sashimi is here in the figure, but they are eating it different way as well. Why they are doing it? One reason is why they are doing it. They want to have this dumb feeling, because if you have a little bit tetrodotoxin, then what happens? Your your face and your lips are becoming a little bit dumb, and you have some, in brain, in face, you have some, excitatory feeling or something, good feeling or interesting feeling. They want to have it. Therefore, it's popular, it's expensive. If you go to markets, most of the toxin is in internal organs and skin. If you go to market, then they are selling it also there, but quite often without skin and organs to minimize the risk of poisoning. But despite of that, the poisoning is still happening sometimes. And there are some anecdotal cases from old times that people were considered to be dead, and then later they found out that they are not completely dead. I mean, it's just tetrodotoxin poisoning, so brain was still a little bit living. Okay, so neurons. So you said at the beginning that they don't regenerate. Neurons have axons also. Axons are more relevant than neuron bodies. If you have a neuron, it dies away, axon dies away, it's dead. If you have a neuron and axon dies away, then this neuron is as dead, because without axon there is no use from that anymore. So the second question is then, which is not so clear maybe, you said neurons do not regenerate. Is it fully correct? Is it fully correct that they do not regenerate? And in the another questions, do the axons regenerate? What do you think?
Branching occurs in every cell. They increase the branching.
Yes, but if you take, for example, motor neurons, then it's one big axon and the branches are only at the end. So if something happens with these axons, can this axon regenerate, grow again? What do you think? Yes, please. What do you think? You are quiet from in this side. Can axons regenerate? if you think about the people who for some reason uh, uh, they are breaking their spine or something, what happens with them later? They are in wheelchairs. And very rarely, sometimes there is some regeneration, mostly not. So that the answer will be that axons don't want to regenerate in most of cases. But there is no clear no here. I mean that usually neurons do not regenerate. Usually axons do not regenerate. So, what is clear that mature neurons do not divide. So they are out from the cell cycle and that's it. So you don't get any new neuron from fully matured neuron. But theoretically you can get new neuron from what kind of cells? Progenitor, neuronal progenitors. You don't have these progenitors very much in the brain, but you have something in hippocampus. So therefore people are speaking about adult neurokinesis. So that in hippocampus, in dentate gyrus, it's very small, this hippocampus here, dentate gyrus, in this red area, you have some progenitor cells, neural stem cells. And these cells are giving us the new neurons. And these new neurons can theoretically migrate also further to CA3, I mean to this hippocampus area, CA3, which is not this dentate gyrus, which is more far. So that there is some regeneration at the level of hippocampus. There is no, and it's not so big, but it's there, because and therefore hippocampus is more plastic, how to say, there is more plasticity. There is no regeneration at the level of cortex. People are trying to use it, because if you take a Parkinson's disease, in very specific small area in striatum, the dopaminergic neurons are dying. So there was a logic that you will inject these stem cells to striatum to get new dopaminergic neurons. Very logical, maybe it helps. But you think, was it helping? No. Because the cells survived there, these stem cells, but they were not giving really neurons. I mean, they did not multiply. So it was not working. And maybe somebody is still trying, but during last 10 so years, it has not gave anything. Then another thing is axonal regeneration. It's also relevant because there are many situations where axon is dying or axon is damaged. So two main conditions. They are not exactly neurodegenerative diseases, but they are important. One is a spinal cord injury. So something happens with spinal cord, you will fall from somewhere and break it. And there are quite a big number of people who have that. So they are going to wheelchairs, they are paralyzed from some place, and this is not moving. And then maybe as well relevant or even more relevant is the traumatic brain injury or intracranial injury, which is when something hits your brain or head too heavily, or you will fall to floor too strongly. This means that your brain inside of the skull is moving very rapidly, and during this movement, rapid movement, it could break some axons. And this axonal breaking means that there is no connection there anymore. The spinal cord injury is usually yes or no. I mean, you have it and it's full, not always. But the traumatic brain injury could be very different. It could be severe, it could be mild, and so on. But nevertheless, it's big a problem. And people are thinking how to do something, how to get axons regenerate somehow. It is not really so easy. So there is also another thing, that sometimes you could have a peripheral nerve injury. So this was about what happens brain and spinal cord, but you can have also axonal issues in the leg or so on, and leg is not moving. So it's in peripheral nervous system there is minimal regeneration. So some cases your axons are somehow crushed here, but you could get improvement, not full but partial. But at the level of brain and spinal cord, you don't get anything. So this few pictures here about traumatic brain injury. It's not just that the axons will break. It could be also that the axons, if the force is somehow like that, then the axons will not fully break. There is just a twisting or so complete. But even completely axons are somehow damaged and die, so it could deteriorate after that. So what people are thinking, because one review we published quite 10 years ago, because people know that actually the axons can regenerate, but there is something with post-mitotic neurons which blocks. There are many different reasons why regeneration is, but we propose lack of the energy of mitochondria damaged axon. Speculation was axon is eating because of lack of energy at the end of. So this has been, it was then just a high paper, then it has shown that indeed will have a better energy at the end, more mitochondria, then it little bit improves axonal regeneration, but no treatment. So do you have evidence regarding this part? Why nerve system and nerve and the neurons are so damaged? Thing I missed. There are a lot of things which are making them also more sensitive. No questions, then I think we have a break then. Okay? Okay to have a break. Good. And how long we will have a...
We can become lunch break.
Okay.
And then we can be back at around 15 or...
Okay, this is okay.
So now, so can I announce the break?
Sorry?
Can I announce the break or
Yeah,
you have something to say?
yes.